So the next class of non-coding RNAs we'll think about are the piwi interacting RNAs or piRNAs. And these are one of the much more recently discovered classes of non-coding RNAs. So they're best characterized in Drosophila and probably in worms, otherwise known as C. elegans. But we do now understand a little bit more about how they function in mammals. And what they seem to be doing is they play a role in silencing the transposable elements. These are those repetitive elements that I've described before, that are able to transpose themselves or jump around the genome. So these particularly involved in silencing the transposable elements. piRNAs are silencing them in the stem cells and in the germline. And this is to be able to maintain genomic stability. So to remind you what we'd spoken about before with transposable elements or repetitive elements was that they can, first of all, they can jump out and place themselves somewhere else in the genome, and this causes disruption, genomic disruption. They also have very strong promoters which can act out into the neighbouring genes and disrupt the transcription of neighbouring genes. And of course, because they're repeats, they're the same as one another, they can cause aberrant recombination between chromosomes that should not otherwise be recombining. But if they can be silenced, epigenetically silenced, this will decrease all of those functions and limit the damage that the transposable elements can cause. So the piRNAs are helping to maintain genomic stability in that way. Perhaps the reason this is more important, or that it happens more often in the germline and in stem cells, is because these are the occasions when the genome undergoes a lot of epigenetic reprogramming, and many epigenetic remarks are removed from the genome. And therefore, the transposable elements, the previous epigenetic silencing, needs to be re-established. And so we think the piRNAs are involved in this mechanism. So they're longer than the microRNAs. These ones are 24 to 35 nucleotides, rather than being the shorter 19 to 24 as for the microRNAs. They are transcribed from the transposons themselves, and they either have unidirectional promoters or bidirectional. They're also transposed not just from complete transposons, but from transposons or repetitive element remnants that are found throughout the genome, and also from clusters of such factors, so clusters of these called piRNA clusters. We know that the non-coding RNA, so the piRNA, is bound by piwi proteins, and in mammals they're known as MIWI1 and MIWI2, or their other name in mammals is piwi-like1 or piwi-L1, piwi-L2 and piwi-L4. The important thing is, while there are many piwi proteins, their roles are non-redundant. In other words, each of the different piwi proteins has a specific role. So I'll explain the same thing again, but go through with the picture this time. So we have the transposons, which are shown up here, and the transposons can have unidirectional or bidirectional promoters, and they can be clustered or separated out throughout the genome. And they result in, when they're transcribed, they result in piRNA precursors. It's these longer precursors that are then exported from the nucleus and then undergo primary processing. And primary processing in this case just means that they're shortened. They're cut down to slightly shorter lengths to be that 24 to 35 nucleotides. It's at this point that those short RNAs are loaded into the piwi proteins. So this can be the three different types that are found in mammals, for example, MIWI1 and MIWI2. So there's a secondary processing step in addition to that primary processing, which is shortening. And this secondary processing event is known as ping-pong amplification. And I'm not going to go through the details of ping-pong amplification, but all you need to remember about this is that it actually amplifies that piRNA signal in the first place. And so it actually produces more RNAs for silencing the transposable elements than were originally produced. So the first option of what can happen now is that these piwi protein and piRNAs can result in post-transcriptional gene silencing, just like happened for microRNA. So in this case, it means that we get degradation of the transposon RNA. The transposon RNA would normally be encoding genes that are helping the transposon jump around the genome. So it's a very good thing if you can disable that by cutting up this RNA and therefore disabling the ability of that transposon to jump. However, perhaps the more interesting feature of the piwi proteins and the piRNAs that bind to them is that they can be imported back into the nucleus and direct DNA methylation. This only happens in the case of MIWI2. So MIWI2, this one particular piwi protein that's found in mammalian cells, can bind to the piRNAs, go back into the nucleus, And now through sequence specificity of the piRNA that's found there, it will go back and bind to a particular transposon and direct DNA methylation there. So it's this transcriptional silencing rather than post-transcriptional silencing, and the DNA methylation that ensues, so the RNA-directed DNA methylation, which is one of these unique features of piRNAs and why there's a lot of interest about piRNAs at the moment in epigenetics. So if we compare piRNAs and microRNAs, we know that microRNAs require DISA, this enzyme DISA, whereas piRNAs do not. We know that microRNAs only act in post-transcriptional gene silencing, but importantly, piRNAs act in both post-transcriptional gene silencing in a similar way to microRNAs, but also they act in this, they can alter to at the epigenetic state. So they can go back and permanently silence the transposon from which they came through DNA methylation. piRNAs are also important in development and disease. As I mentioned earlier, they're important in the germline and also in stem cells. But in addition to, and we can see the role in the germline by knocking out of these various pIRNA proteins in mice. So knockout mice that are deleted for either MILI1 or MILI2 are sterile, and that's because they have impaired spermatogenesis. Oogenesis, so the production of eggs, is actually fine, but the production of sperm is compromised. And this is associated with a failure to silence these repetitive elements. You'll remember I may have mentioned right back when we were talking about DNA methylation, these intracisterna A particles, or IAPs. It's one class of repeats, a common repeat that's found in the mammalian genome. There are also line ones and many others. And inappropriate activation of those repeats is associated with infertility in these mice. What's important is that if you find people that have mutations in these pIRNA proteins, they are also infertile. And so clearly this is a broader mechanism. Furthermore, if we look in cancer cells, we can also now find that the pIRNA proteins are aberrantly expressed in cancer cells. And so this suggests that they're having a role not just in the germline, but also in other contexts. And it's possible that it's the role for pIRNA proteins in cancer has some relationship to their role in stem cells. This is something that will be investigated or is currently being investigated at the moment. So I've mentioned that these piRNAs are really critical for silencing the transposable elements, the repetitive elements. But do they have a role at silencing other areas in the genome, at non-repetitive regions? What we know is that when you look in a particular type of spermatocyte, we can find the piRNAs are targeting not just the repetitive elements, but also non-repetitive DNA. We can particularly look at one gene, and it's called RASGRIF1. So RASGRIF1 is quite a special gene because it's subject to genomic imprinting. What this means is that it's only expressed from one allele. For all of your genes, you inherit one allele from your mum and one allele from your dad. And normally, both alleles would be expressed. You would use both of them. But in some particularly special cases, you only express the copy you inherit just from your mum or just from your dad. In the case of RASGRIF1, it's paternally imprinted. That means it's silenced on the paternal allele, and it's active only from the maternal allele. So you only use the copy that you inherited from your mum. And so this very special class is called genomic imprinting, this class of genes that undergo this monoallelic expression based on the parent of origin, and we'll cover this in later lections on imprinting. But here what's relevant is that this is not a repetitive element. It's not strictly a repetitive element. It's a single copy gene, and we know that piRNAs are involved in epigenetic silencing at this single copy gene. And so while piRNAs are still only very recently being studied, it would appear that that there's still much to be understood and that they may be acting at many more places than just the transposable elements in the genome.